Hi students, let's start the classes again. Do not worry about your attendance. This should not be your reason for worry. I'm here for you, and of course you will only get the part of your subject in your exam that you would have done. So do not worry about things like these. Try to learn uh, whatever we discuss in the lectures, because you will get all the questions from the parts that we cover. Now, you're a student of pharmacy, and it is very, very important for you to learn about COVID-19. Not just because you can provide the right information to your friends and family, but also because this is not going away in a day. I guess many of you will finish and join the industry, join the vaccine making manufacturers, or even get into the research, and maybe you are still researching on COVID-19. So it is not going to hurt you to know about this. And for this purpose, I'm making it a part of your syllabus. You will get questions from this part also in your exam. I've been a little bit ill, and I don't know if I should say little bit, I've been very ill, and now things are getting better. I will try to do lectures, more lectures according to the university that is online right there. But maybe I will do some lectures which I will send to you through WhatsApp. Make it fun for yourself. Listen to them when you have a little bit of time, late in the night, morning, whenever. We will not push you too much on how much syllabus is covered, but you must go through these lectures because they are part of your syllabus. So I'm starting this with this heading, Pharmacy Graduates, Do You Know? And believe me, I'm doing this for a purpose. A lot of people who talk to me, they seem to know not much about this. They believe whatever they see in the news, and I really hope that as pharmacy graduates, you're not doing that. You're putting your pharmacy mind and trying to make head and tail, and the right head and tail, out of all this information that you're given every day. So before we start learning about what is coronavirus and what all you should know about it, we should, as students, know about CDC. CDC is Center for Disease Control and Prevention, and every country has these CDCs. They work together with each other, and they have defined three types of these diseases like these, right? CDC defines these infections as outbreak, epidemic, and pandemic. Now, the question is, do you know why it is called pandemic? Why COVID-19 is called pandemic? What do you understand by the word? So we are going to reach that. Outbreak is when the disease is reported in numbers, quantitative numbers, in a given area. So not all over the world, but just in a given area. You see that a lot of patients are coming into hospitals during a particular period. This is called an outbreak. Many people, 100 people are coming into in 2 or 3 days in a hospital, for example in Wuhan, and the doctors are not able to categorize the disease, but they know it is something because 100 people came in. Such a thing is called a outbreak. Okay, so when it happens, it's happening in a certain area. Epidemic word is usually not used. it could also it actually leads to panic, because epidemic means that people are dying in high numbers, high mortality rate. So it is not used, and it usually means a broader geographic area. For example, China has it, okay? Not the rest of the world, but China is suffering from a epidemic, right? That's an epidemic. Like a black plague was an epidemic that was in the area of Europe only. Pandemic is when multiple countries or continents get involved. So if you remember, not in the starting, but somewhere in January end or February or even later than that, I can't remember, maybe in the next slides I have it, when COVID-19 was found in more than 50 countries or something, it was declared as a pandemic. So COVID-19 is now a pandemic, and it obviously goes through the stages of being an outbreak and then becoming a pandemic. So we'll go through these important questions. How was this outbreak identified? Coronavirus outbreak. What pathogen is causing the outbreak? Where did this virus come from? Now there's a lot of shit, honestly, it is all shit that is being reported in the news, and I want you to think like a pharmacist and think about this. No, people were not getting it from eating bats, no. So you have to process the information that comes in the news very, very carefully. How dangerous is this new coronavirus? We'll talk about it. I think the information will get more and more updated in the time to come, and the high mortality rate, especially in Italy, there are many other reasons than the virus itself. What is the life cycle of the virus? This is very important for you as a graduate for your course, because this is how you would make medicines, right? You would target different life cycles of the virus and make a new medicine. And what can be done to keep it from spreading further? This is a debatable thing, but I still want to tell you in one slide so you can protect yourself. And this is going to take time. This COVID-19 is going to take the time to get over. So be prepared, and prepare your family also with you. Alright, so if you go through the WHO's report, the main site or the link that I've provided you in this deck of slides, you'll see that the first report started coming in December 2019. Please do not believe anyone who says China did not tell the world and whatever, because I don't think it is true. If you look at the timeline, you would realize that they worked very, very efficiently, very fast, knowing that a new infection or outbreak is happening, to telling the whole world. And you have to remember, people are coming every 2 weeks, so it is a very difficult process to realize. What we can say is that the first report came in Wuhan in December 2019. It spread very quickly. So by February 16, over 50,000 cases were confirmed in labs using kits. I will also tell you what kind of kits these are. And more than 1,600 were dead. So if you go to the ARCGIS site, ARCGIS site for WHO, you can actually see every day reading about how many people have died and how many cases, and you would realize there's a jump in between. This is because especially in the detection, this is a jump because as soon as new cheaper kits were available, more people could be tested, so limited more by the kit availability. The mortality rate is unfortunately does not change much. It is going on increasing because people have been affected before the isolation started. By March 11, so this is the date, March 11, it was labeled as a global pandemic with more than 100 countries affected. I think this was the problem, that even if we knew that such an infection was spreading, we were not able to limit the globalization and limit it to a certain area because of transport, because of the business going on between different countries. Unfortunately, many people have died till now. The numbers are from March 11, 2020. So this is an image from the CDC site from March, and you can see all these different countries in blue. These are all affected by the pandemic, India included. I think the countries where we do not see any blue, the main reason is either they are very poor countries, they don't have access to kit, not at the time when this image was made. Mostly African, Kazakhstan, all this area. And also because maybe they don't have much of a business going on with China. So how was this outbreak identified? It started in early December 2019. Several patients came into Wuhan hospital. You have to imagine it in your brain. They come into a hospital and they get admitted. The doctors don't know yet about the infection capacity of this virus. They don't even know it's a virus. But they know it's something different. It's maybe a new kind of flu. They're not wearing any gloves. They're not taking the precautions that are told now. None of the other patients are doing that too. None of the family members are doing that too. So in the end, this infection or this flu is labeled as pneumonia of unknown etiology. Now remember that this phrase, pneumonia of unknown etiology, is actually developed after the SARS outbreak of 2003 to flag for public health officials that there is a new type of pathogen. So whenever you read something like pneumonia of unknown etiology, it should tell you, oh, there's a new type of pathogen. This is for the public health officials, doctors and people who make medicine or who make the decisions regarding new diseases. So that if it is like in case of coronavirus, if it has high mortality, things can be properly dealt with. In the next 2 months, the danger around it became clearer. On the last day of 2019, so early December to last day of 2019, WHO China country office. So every country has a WHO office, and the WHO China country office reported it as a potential public health concern. Okay, so now the WHO also knows that this is a potential public health concern, but it's a potential, right? This is quite fast, like in a month of this, WHO already knew that it's a potential public health concern. You have to know, people are developing it slowly, right? And not like in a day, but it takes 5 days to show the symptom, maybe 3-4 days more to get really, really ill, and not everybody is falling that ill. In early January, cluster of patients came in. Okay, this is how it was defined as an outbreak. So a cluster of 44 patients came in with the pneumonia of unknown etiology, and therefore it became an outbreak or epidemic. On January 30, okay, so 2 months have passed by now, WHO declared it to be a public health emergency of international concern. So it is still not labeled as pandemic and people are going around doing their daily business. But WHO has declared it as a public health emergency of international concern. And the name COVID-19, 19 for 2019, because the virus infection actually started in the end of 2019. So what pathogen is causing the outbreak? So believe me, this is quite fast. In December they get to know, and in early January, so the doctors are already isolating the pathogen. Coronavirus is isolated and identified. How do you think this happened? How did they identify it was coronavirus? There's a reason. All these are PCR based techniques. So there is a difference in COVID-19 from the previous coronavirus, but you don't have so many differences in the genome. You can still see that it is a coronavirus. If you use PCR based methods, you can still tell by the sequence similarity that it is a coronavirus, right? Coronavirus are a family of enveloped viruses. So if any of you have been going online and searching for yourself, you always see a image of a virus. I'll also show you in a second. So it's an enveloped virus, and what are viruses? They are just envelopes, and they have some kind of DNA or RNA in the inside of this capsule. Coronavirus has positive strand RNA genomes. Okay, so this is inside that capsule. And it is not a new virus for outbreaks. There have been many viruses before this. Corona family viruses such as SARS in 2003 and MERS in 2012 have already been involved in outbreaks. So it is a family which has already been studied quite a lot, right? During these outbreaks, a lot of information is available. So there's no need to panic. So here is a typical structure of coronavirus. On the right side, you see this is a capsule. You cannot see the DNA or RNA in it because it is inside the capsule. You see these red spikes. These are the spike proteins on the virus, and the virus uses them to gain entry into your cells. The name that has been given is SARS-CoV-2. This is because it has high similarity with the SARS-CoV 2003 virus. So they have given it the name SARS-CoV-2. It is genetically distinct. How would you call anything genetically distinct? It has some mutations in this RNA strand genome that are different from the previous coronaviruses. That is how it is genetically distinct. Okay, this information about low apparent case fatality rate and higher transmissibility rate is from March 2020, and it is very difficult to tell you the exact fatality rate right now because these studies are usually done when the outbreak is either at a constant or is over. But this is for sure true that there is a higher transmissibility rate. I'm actually amazed as a scientist at the rate. It's really amazing. I've never seen something spread so fast. Countries going into quarantine, and I would be very willing to work with one of you on finding out why there is a higher transmissibility rate than SARS-CoV. But believe me, this is the reason why we are in quarantine, because the transmissibility rate is high. And many people, around I think 80 to 90%, they survive. So the fatality rate is not so high, but the transmissibility rate, I don't know how this is happening. I mean, if one person sneezes, right, and it's in the air next to you, it would still it should not still be so high. Anyways, we can discuss this at some other point of time. So this virus is called SARS-CoV-2, and in the patients that are severely ill, severe acute respiratory syndrome is seen. Where did SARS-CoV-2 come from? So it seems, because we cannot identify the patient 0 here, it seems that it is in early December 2019, and evidence suggests that it is possible that there were people who were sick before that too, but we could not identify these patients. For the family, coronavirus family, it is well known that the route through which the infection gets transferred to humans is called zoonosis. There's a word for it, zoonosis or zoonotic spread. There were a lot of news in the starting which said that this comes from the Huanan seafood wholesale market with some contaminated seafood. But now we know that it has high similarity with the coronavirus found in bats. Now, don't jump to conclusions. This does not mean that the Chinese people were eating bats and that's how it spread. No, for this you need to go and study more about it. What is a zoonotic spread? This I can tell you. When viruses mutate and change through many rounds of replication in their animal host, such that some of the virions acquire the ability to infect new host. This is a zoonotic spread. Not that Chinese people were going and eating bats. It's the virus itself that mutates by many rounds so that it acquires the ability to infect new host. So just as when a human being is infected with a virus and they sneeze, and the sneeze causes infection of new humans, it's the same way, right? What are the other examples of zoonotic spreads? The avian flu, the swine flu, all of them crossed over from bird to human or pig to human using zoonotic spread. So this is the word you're looking for, right? Now, how dangerous is SARS-CoV-2? SARS-CoV-2 or COVID-19 is different from SARS, okay? The symptoms are a little bit different and even the severity is different. But it's very close, right? So 2003 SARS virus had severe respiratory symptoms and the fatality rate was around 14 to 15%. While COVID-19, the symptoms are different. Majority of people have fever, cough, and relatively mild symptoms. So many a time we are not able to tell if this person has COVID-19 or not before you do the test. And even if you do the test, it does not mean that this person is going to get severely affected. Maybe their immunity just fights through it and is all right in a few days, I think 2 weeks. SARS-CoV-2 has an incubation period of 5 days in most of the people, but some unlucky people like who has autoimmunity disorders or have a weaker immune system, you can even see an infection in 1 day and maybe even more severe symptoms. So there is a difficulty in estimating the mortality rate, and maybe in a year we will have the right idea about how many people are actually dying from SARS-CoV-2. Direct fusion with the membrane, the RNA is already released. But when it is through the ACE2, then the RNA is released upon entry of the virion into the cell. So the third step is RNA release. This RNA is coated for protection in the virus. So now if we want to translate this RNA to protein, first it has to be uncoated and it releases a plus strand of RNA, right? This gets translated and polyproteins proteolyze this short protein to give these I don't know how to say them, but these infecting or these replicases enzymes that then cause this positive strand to become a negative strand. Why do we need the negative strand RNA? Because until unless it is a negative strand RNA, we cannot or the virus cannot replicate properly. Okay, so the positive strand has to be converted into a negative strand, right? So, and the next step is obviously therefore the transcription of this negative strand RNA and RNA replication using the host machinery, right? Once it is transcribed, the negative strand is transcribed, then it is translated and at the endoplasmic reticulum. Now you have to realize one thing that we notice here is that the RNA from the virus did not enter our nucleus at any point, any cell's nucleus. So this is, I think, the advantage on the coronavirus part, that it does not need to enter our nucleus. As soon as it enters the cell, it can go on and get translated, use the machinery of endoplasmic reticulum and the Golgi apparatus, right? And if you remember your human anatomy and physiology lectures, then endoplasmic reticulum would lead to the production of protein, translation, and Golgi apparatus puts any of your proteins into a vesicle which can be transported to the plasma membrane, right? So the virus uses the same machinery to make its virion, right? To put the positive strand RNA into the viral capsule, and then this viral capsule is put into vesicles, which is the normal way how our protein also reaches from Golgi to its destination. Here it's the plasma membrane, right? Maturation and budding of vesicles and translocation and fusion of the vesicle to the membrane so that the virus is released. So this is the life cycle of a coronavirus in your cell, right? So there are different regions which can be targeted during therapy, right? Now the most important question is, bat is not dying from any bat is not dying from coronavirus, but this virus comes into a human host and humans are even dying. They are also showing symptoms. Why is this? Why are we not dying, right? Why are bats not dying, right? Why are bats not showing any symptoms? Sorry for that, right? Okay, so the most important thing is when the virus enters our human cells, it is in the cytoplasm, right? Remember the human anatomy and physiology lectures? This is a cytoplasmic piece of DNA. Now this is not very common. There are only two regions in your cell where DNA should be. What are those regions? Nucleus and mitochondria, right? We don't have any floating cytoplasmic DNA of alien nature in our cells. So of course, our human cells have a mechanism to deal with it. If any such cytoplasmic DNA enters a human cell, it is sensed, right? Like a sensor. It is sensed by a cytosolic DNA sensor protein called C-gas. This C-gas protein senses, there is cytoplasmic DNA, right? And it activates a protein called STING. Now you have to remember that whenever we say activation, inactivation, inhibition, all this, activation of many proteins is actually done by phosphorylation of that protein. It is phosphorylated. It's a chemical mechanism, right? So the STING gets activated by phosphorylation of S358. Alright, this is the serine at the position 358 on the protein which gets phosphorylated. Right? This in turn phosphorylates other STING proteins as well as IRF3, interferon factor 3. As soon as you hear interferon, inflammation should come to your mind, right? So you have to imagine it like this. Cytoplasmic DNA, C-gas senses, oh my god, there's cytoplasmic DNA, phosphorylates STING, which then phosphorylates many other proteins, many IRF3 molecules. Interferon levels increase in your cell, indicating inflammation. So one of the first indicators of any such infection is inflammation, right? What happens in bats? Why don't they show anything? Now bats have low inflammation because of their unique virus fighting mechanism. This also comes from their continuous flying. Because they are flying continuously, they get subjected to much higher levels of cytoplasmic DNA. When their C-gas senses this, it does not completely activate STING because the S358 mutation happens. So this S358 is lost. There is another amino acid. Therefore, not a lot of IRF3 is phosphorylated, maybe a little bit, but the inflammation levels are low. So it is possible that these bats which had COVID-19, they were in the environment for a long time, but we could not tell. The bat researchers could not tell because they don't show any inflammation. So this is the main mechanism why you're seeing it in humans, because we have a very good mechanism to sense this cytoplasmic DNA in our system, and the inflammation is the main reason why we are seeing so much sickness, because more the cytoplasmic DNA, more inflammation of your cells is being seen. Now go back, I actually would like you to go back and look at the symptoms and also look at what is happening at the level of your lung cells, what is happening. And you will see that inflammation plays a huge role in, especially in patients which develop serious problems, right? So what can be done to keep viral outbreaks from spreading? The first thing is rapid detection. You have to detect it very rapidly, right? And this is what has been in the news for the last few weeks, because now we have a kit thanks to one of our scientists, which is much cheaper, and therefore you see such a huge detection, right? You saw a jump, not because patients were not present before the last 2-3 days. They were always there, but we did not have enough kits. And now you see a lot of people being tested, and the government is able to tell if they are positive or not. So you see a huge jump of, I don't know, 600, from 300 to 936 or something, if I'm not wrong. So the reason is now we have diagnostic lab kits, and of course, think about it. How would the kit work? It has to work through PCR. It has why PCR? Because you want to detect the cytoplasmic DNA, right? The RNA, right? Because that is the only evidence that the virus is in your system. Inflammation can be from any reason. So to detect this RNA genome, you have these PCR kits, and if the patient has the RNA, we can detect it through selective primers, because the primers are specific for the RNA sequence of COVID-19, and we humans don't have the sequence. So a patient will only show this if they have the virus. So what are the things you should do as a as a aware person? Wash hands frequently. Definitely wash your hands frequently, especially if you're you know that you have been outside home, or you were somewhere where somebody could have been infected. Practice cough and sneeze etiquettes. Practice these things, right? You sneeze into the elbow, or and dispose of used tissue. I would really suggest keep a little bit distance from everyone in your family, right? Not just because you want to be safe, because you want to keep them safe. And we don't know why, not yet at least, why different people are reacting in different ways. It would definitely not help you if your mother is sick, right? Because she is kind of managing your whole house. So if you feel you're falling sick, I would really suggest that you be careful about who you are coming in contact with. If you're wearing any face mask, be very careful, because I don't know about you, but I have seen that in my locality, the people who are cleaning and who are taking away our garbage are still working, and if you do not put or separate this kind of garbage and make sure that they don't get infected, this can be very, very tough on the society. So be careful if you wear anything like this, or even these used tissue. Make sure that they are properly disposed of and not out in the open, right? What I want you to do after this class is I want you to go ahead, right? And if you felt like you want to know more information, I want you to go and look at these questions online. These are the questions that will help you also for your future, whatever you're going to do in the future. Because COVID-19 will take a little bit of time. If we are lucky, the vaccine will be available in, I don't know, 1 year. But you still want to read about these things. Look at scientific papers on what type of drugs will be useful in this infection. Look at the scientific papers, not at the news, right? Look at the scientific papers. What is the newest information that came out? What kind of drug can be used in this infection? Is it a vaccine? If it is a vaccine, how will this vaccine work, right? What can you do to take care of yourself while scientists find a drug, right? And what are the scientists saying? What are these scientists saying? What is ICMR saying? What are the government agencies saying, right? what is WHO saying? Look at all this. Help yourself, right? Help me. And I hope you enjoyed this lecture. I'll try to do this till I get completely healthy, right? And I wish all of you health too. Take care of yourself. Don't worry about your attendance. Don't worry about the exams. Um, they will only be from the part that you get taught. There will not be any extra burden on you, right? Take care. See you soon.